Welcome back, Mindsetters. We are doing life sciences and it has been absolutely awesome because it is revision, and revision is always good. I mean, always good, because you can never know too much, really. If you do want to pass those exams, especially for those of you who want to get exemptions, this is a very important show for you. And I'm glad that you guys have joined us and you've been absolutely focused because the questions are absolutely amazing as well. Aslam, there's an interesting one.
Yeah.
Mutati says a good one is good couples always together for GCAT.
Good.
That is awesome, hey?
Good couples
Always together.
together. There you go, guys. You can make your own mnemonics. TACG. Good couples always together. There, I've learned something great today,
Yes,
and I'm
that's
going to
awesome.
definitely. What's the name again?
Mutati.
Motlatsi, thank you very much. I'm going to be using that. I hope you haven't patented it yet because I'm going to be using this one. Good couples always together. There's your G with the C, A, and T. Great. Guys, in this way, you make the mnemonic that makes sense to you. Whatever makes sense to you. We're giving you guidelines, so great for that one. Any other questions there?
Yes, we do have questions. We have one by Sfiso. He asks, what's the difference between monomers and nucleotides?
All right, let's look at these two words. Monomers and nucleotides. Fiso, a monomer is a building block. Much like a brick in a wall. The brick is a monomer. The single smallest unit that a wall is made up of is the brick, and the brick becomes the monomer. In DNA and RNA, the nucleotides are monomers. These are not synonyms. They're not synonyms. A monomer is a building block, but like the brick was a monomer in a wall, the nucleotide is a monomer of a nucleic acid. A nucleotide, by the way, is made up of a phosphate, a sugar, and a nitrogenous base. For example, in this case here, this is an adenine nucleotide. And it's a building block of DNA. So therefore, it's a monomer as well. So the word monomer means building block. A nucleotide is made up of phosphate, sugar, and nitrogenous base, and a nucleotide is actually the smallest part of a DNA, and those are the monomers of a DNA.
Okay.
One more question there?
Yes, there was one more by Vuyelwa. Vuyelwa asks, what are the differences between lethal, neutral, and fixed mutations? So that's lethal. Vuyelwa.
Vuyelwa, lethal.
Oh, sorry, Vuyelo.
Vuyelo. Okay,
yes.
good. Vuyelo wants to know about lethal, fixed, and neutral. Lethal, the lethal weapon. What's the guy's name again? Mel Gibson.
Yes.
Lethal weapon. Lethal weapon, a weapon that can kill you. A lethal mutation is a harmful mutation that leads to the death of an organism. That means it's so bad that it's going to kill you. That means you cannot survive with that mutation. Straightforward. We go from there right to neutral. Neutral, guys. If I tell you I'm staying neutral in this matter, or if the car is not in drive or park or first, second, third, fourth, fifth, or reverse, it's in neutral, it means it's in no gear. So neutral means it does not have no effect actually I should say, no effect on the organism. It is there, but it's not going to make a difference to the organism. And a fixed mutation is generally one that becomes beneficial. That this mutation is so good that it gives certain organisms advantages over other organisms. And therefore, these organisms survive, and the ones without this mutation, they die. So this becomes a beneficial one, and it stays with the organism. So it's a fixed mutation. Simple explanation, guys. Don't look into too deep into this. Just remember this what I have explained here. That's all for now?
That's all for now.
Great, guys. Great. Keep bringing those questions in. Now we are moving on. We've covered a bit of protein synthesis. We've covered our DNA and DNA replication. We've covered some of the mutations. We're moving on to technology now. Gene is a segment of DNA with bases in a certain sequence. Human genes can be isolated and then inserted into bacteria. The figure below shows how insulin can be synthesized by inserting the human insulin gene into a bacterium. After this has been done, the bacterium is known as a transformed bacterium or a transgenic because we have taken a gene from one organism and put into another organism. This is just one example of that. So they're giving you the whole story here. By the way, this one here we've taken from one of the high schools in the Western Cape, and we've adapted it for ourselves here. Great question. Normally we get a diagram that's fixed here. They've made their own diagram. There's a human cell. Here they've taken the chromosome from the pancreas of this human, have isolated it. They have cut that insulin gene. At the same time, they get a bacteria. They take a plasmid. They isolate it. They've taken a plasmid and they've isolated this plasmid and they've cut it. Look at this diagram here. Look at this diagram there. They have cut it using an enzyme. These enzymes are called restriction enzymes. Restriction enzyme. By the way, this enzyme works like a scissor, not a physical scissor, but a chemical scissor that breaks this plasmid and leaves a spot there. Now what they do, notice what they've done here, they've isolated the DNA, they've taken out the insulin gene, they've cut it, also the same process, restriction enzymes. They've taken this enzyme, this gene, and inserted it here, also with an enzyme. And they then take this whole plasmid and put it back into the bacteria. Now what do we have? We have a transformed plasmid into this bacteria, so we have a transformed bacterium. They allow the bacteria then to reproduce. And these bacteria produce more and more of the same. They then remove the insulin that is formed, and they store this insulin. And this insulin then is available to diabetes sufferers in an injection form or in a vial, a little tube where they can inject themselves. The first question here is, what is a plasmid? And obviously the diagram is telling you that. What is a plasmid? It is a DNA, so it's a ring of DNA found in the bacteria. What molecules is the insulin gene made up of? What is it made up of? And in this case here, obviously these two questions wouldn't come the same in an exam situation. I've taken it from different papers. And we answered that a little earlier, that insulin is made up of protein. The last question gave you that. Okay? What would we call the four identical cells at the bottom of the diagram? These four cells that came from one mother cell. They are identical copies. And if we use this process to make identical copies of organisms, we call the resultant organism a clone. Clone is a copy of a cell, or of an organism for that matter. We're carrying on with the same question. Suggest what type of reproduction occurs in the transformed bacterium. Now, they do not expect you to know the specific type. And if you do reproduction, you know you learn two types of reproduction, asexual and sexual. They want to know which of these two would it be. And you would say asexual. By the way, just for our knowledge, the actual type of reproduction, the example of asexual reproduction that takes place in a bacteria is known as binary fission, where one cell, by mitosis, makes many cells. So the one cell divides and the chromosomes go to opposite cell. The cell pinches in the middle, we get 2, 2 make 4, 4 make 8, and so the story goes. But it's asexual. Suggests why this type of reproduction is needed for the large-scale production of insulin. We want insulin. What insulin? The insulin that is found in humans. We want to use that insulin. Can sexual reproduction take place here? No, because when sexual reproduction takes place, a variety is introduced. So why do we need this? Because we want identical copies. of the insulin. We do not want a variety here. In this case, we do not want variety. In this case, variety is no good. We don't want to change it. We want it the way it is. That insulin must do the work for us. Explain 2 advantages of producing insulin from bacteria rather than obtaining the hormone from the pancreas of an animal. Now, in this case, they can ask this question both ways. They're asking here the advantages of doing it this way. Or they could reverse it and say, explain 2 advantages of obtaining the hormone from the pancreas of an animal as opposed to getting it from human insulin. Can you see how that can be done? So obviously, you've got to take this disadvantage and make an advantage, or the other way around. And what are the advantages? First of all, if we needed the pancreas of an animal, we needed to kill that animal to get it. So therefore, we're going to avoid large-scale killing of animals. Okay? But notice what they said there. Oops, sorry. Notice what they said there. They used the word explain. Why? Because you must give a reason now. Why does it do that? Because we use human insulin. Okay? The next one would be, obviously, there are fewer or no side effects. Because, again, we're not using animal insulin. And thirdly, there may be no religious objections as we're not using animals. Okay, certain religions have objections when you use products from an animal. For example, myself, I cannot use any product from an animal that hasn't been slaughtered correctly. I cannot ever use, whether slaughtered correctly or incorrectly, any animal that is, well, not any animal. I cannot use any pork, pig byproduct at all. So different religions may have different objections to that. So therefore, this way, we're not using an animal, we're using human insulin, and therefore, there's no need for religious objections. I've just given you 3 there. I'm going to have to leave this question out. We wouldn't have enough time to do that one, so we will have to do that some other time. I just want to go to this last one here, because this is just a discussion. It's not going to go into details of the answer of this question, because we've covered most of it already. I'm going to take the core, and we're going to break it down. I'm only putting this because it's an essay type of question, and we need to learn the skill. Somebody asked me on WhatsApp yesterday, one of my Star School learners asked me about the same thing. And I've explained it on WhatsApp. I'm hoping she's watching today, and maybe, not this question rather, but just about writing an essay. So they say, describe how proteins are synthesized. Explain the difference between a point and a frame shift mutation, and the possible impact of these types of mutations on the formation of proteins. So in effect, guys, you need to know how many parts there are to this question. And in this case, there are 3 types. First of all, how proteins are synthesized, 1. And what are we looking for? We're looking for the verbs. Whatever comes after the verb is what you have to do. Describe what? How proteins are synthesized. So in other words, you need to describe protein synthesis. Okay? Explain the differences between what? A point and a frame shift mutation. That's the next one. And the possible impact of these types of mutation on the formation of proteins. So there are 3 headings here that you can put in. So you start with protein synthesis. And I'm just showing you a plan. I'm not going to explain the whole thing. We do not have time. Protein synthesis will be broken up into transcription, which you will explain, and translation. Okay? From there, you will talk about the point and a frame shift, which we've already done. What's the difference between a point and a frame shift? And the last part, you will talk about if a point mutation takes place, how does this impact on the formation of proteins? And if the frame shift takes place, how would this impact? Now, generally, if you look at the point shift, you know the point shift. Look at me what I'm saying. Point mutation. The point mutation affected only one nitrogenous base. It did not affect everything. If it does not affect the type of amino acids in a big way, then it may not have a serious impact. However, if it messes with an important amino acid, it can cause problems. Okay? Whereas with the frame shift mutation, just by adding or deleting one of the nitrogenous base, the whole sentence is wrong. And therefore, the DNA, if it happens to the DNA, it's going to happen to the RNA and so the protein. And with that, guys, I will leave you in the capable hands of
Thank
Bertha.
you, Aslam. What an awesome show. Thank you for that. And thank you guys. You know, Aslam, there's a Facebook message here by Danessa. She says, Aslam is the best life science teacher ever. I understand
She's
everything
lying.
he teaches. Aslam rocks. She's lying.
She's lying, she's
I
lying,
think
she's
she's telling the truth. Many of you feel like that, I know. That's why you all joined us today. Thank you for an awesome show, guys. From me and Aslam, thank you, guys, Mindsetters, grade 12s. You guys are writing exams soon. We wish you all the best in the world, and we know you'll do fantastically. Join us again next week for more life sciences. Bye, guys.